Okay, in this part, first I want to show you how the sarcomere work and how the thin filament and thick filament are located near each other. Now, please watch this video.
Actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins slide toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
So this is the structure of a sarcomere, which I talked about it before for you. And now I want to continue this part. This is the close-up looking of neuromuscular junction. In this close-up looking, you can see the synaptic knob here, and this synaptic knob contain vesicles which are full of neurotransmitters, and you see that we have many calcium channels, voltage calcium channels which become open when the action potential reach the synaptic knob. The calcium is joined with this synaptic vesicle and help this vesicle to do exocytosis. It does exocytosis and the acetylcholine release in the synaptic cleft. Then it attach to postsynaptic membrane and make action potential in the postsynaptic membrane, which is our skeletal muscle here. And this skeletal muscle is called motor end plate in this picture. You see, we have many receptors. When the neurotransmitter attach to these receptors, the channels become open and sodium ion come inside the cell. In the next slide, you can see bigger picture of the muscle fibers. You see that we have neuromuscular junction here, and the action potential which is created here can move and can propagate in the sarcolemma or plasma membrane of the muscle fiber, and it can make depolarization in different parts of our sarcolemma. You know that when the action potential is created, it's very similar to our neurons, to our axon. First, we have depolarization. That part become repolarized and then go to the resting phase. And the next part go to depolarization. So step by step, depolarization can propagate on the sarcoplasmic membrane, as you see here. The important thing which you need to know is this. The resting membrane potential in skeletal muscle fibers is negative 90 millivolt. It's different from the nerve cell. If you remember in the neuron, the difference between inside and outside of the cell is minus 70 millivolt. But here we have minus 90, so our skeletal muscle is little more negative than the nerve fibers. So this is about the resting membrane potential. In this picture, you can see the myofibrils. The myofibrils are wrapped by the sarcoplasmic reticulum, and all of these green structure can show you the T-tubules. In this picture, you have more details. In this picture, as you see here, we have creation of action potential, then propagation of action potential, and then the action potential can reach the T-tubule. When the action potential come to the T-tubule, it can make the calcium channels in the wall of our sarcoplasmic reticulum become open. The calcium ion release inside the cell, and this calcium calcium can attach to TNC of troponin, and then I will show you what happened after that. When this calcium attached to troponin, the troponin start to pull the tropomyosin, and when the tropomyosin remove from the active site of our actin, then the active site become naked. Active site of actin and head of myosin can join together, and then we have a sliding over of these two filaments together. I will show you some videos to understand this part better. Again, in this picture, you can see the nerve fibers, you can see the myelinated axon and the axon terminal or synaptic knob here, which is full of neurotransmitter, release of acetylcholine, and attachment of acetylcholine and our receptors here. In the next picture, you see that we have opening of sodium channels. Sodium ion come into the cell and inside the cell become positive. This is the meaning of depolarization. And in the next slide, I want to show you the wave of action potential which happen in the sarcolemma. First, you need to know that, as I told you before, the differences between outside and inside of the cell membrane is negative 90 millivolts in the muscle fibers. Then the sodium ion enter our cell, inside the cell become more and more positive until we reach the threshold level. The threshold level in our muscle fiber is negative 65 millivolts. So again, it's different from nerve fibers, which is negative 55 millivolts. When we reach the threshold level, the next step is depolarization. Depolarization means inside our cell become positive, and the maximum positivity which we have here is positive 30 millivolts. When we reach this potential, we have some resistance inside the cell which doesn't let more sodium ion come inside. Then the sodium channels become closed and potassium channels become open. Potassium ion leave our cell and inside our cell become more and more negative. And this is the meaning of repolarization. We have a small amount of hyperpolarization here, and after that the sodium potassium ATPase start to work and bring the membrane potential to the normal limit again. In this picture, it wants to show you a process which we call it excitation contraction coupling. In this excitation contraction coupling, you can see the propagation of action potential in the sarcolemma. Then this action potential come and enter the T-tubule. When it enters the T-tubule, it stimulates the voltage sensitive calcium channels, and then they trigger opening of calcium channels in the wall of our sarcoplasmic reticulum. So calcium ion come out of sarcoplasmic reticulum and go inside the cell. This step is called excitation contraction coupling. The next step is called crossbridge cycling. In the crossbridge cycling, the calcium ion, which is released from sarcoplasmic reticulum, come and bind to the TNC of troponin. The TNC of troponin and calcium attach together. The shape of our troponin change, and this configurational change can move our tropomyosin. When the tropomyosin move, then the active site of the actin, which you can see them with these dots, these black dots on the globular actin, become naked. And then when they become naked, the head of myosin and this active site can join and attach together. When they want to join together, we need to break down one ATP. And this breaking down happen by the head of the myosin because the head of the myosin have this TPase activity. I put some videos on your blackboard. These videos cannot play it here. So don't forget to check these videos on your PowerPoint. You have 2 or 3 videos. All of the slides which I pass are videos and you should watch them yourself. And here I want to talk about this cross-bridging. I told you that this sorry, I should start from here. You can see the calcium ion and the TNC troponin join together. The active site of actin become naked. Then the head of myosin attached to it, and one ATP is broken. When one ATP is broken, this angle, which I told you is like the hinge joint, go to flexion. So it starts to move, and this movement means a sliding of actin and myosin over each other. And after that, this head of myosin release from the actin here. They separated from each other, and when they become separated, again the other ATP attach myosin head. It's broken down to ADP and phosphate and bring back this head to its normal location. So you see that during one contraction, break down to ATP. Because of that, for contraction of muscles, we need many ATPs when we want to do workout, when we want to have some activities. So this is the process which happens in the head of myosin and between actin and myosin together. Please turn off your microphone. Amiral, please turn off your microphone. Here you can see the sarcomere. This is the sarcomere during relaxation, and you can see the A band, you can see I band on both sides, and you have your ZDC on two sides. And this is the electromicrograph. You have dark band in the center and 2 light parts on both sides, and you can see a clear H zone with the M line in the center. But when your muscle go to contraction, you see that your actin and myosin overlap each other. Then you don't have any part of your myosin or thick filament which is naked. All parts are covered with the actin, and the length of your sarcomere decrease and decrease. And here you can see the decreasing in the length of sarcomere. In the first picture, this is the length of your sarcomere, and in the next one, this is the length. So you see that it become smaller. We don't have any light band here. We don't have any H zone in this part, and our sarcomere become shorter. This is the meaning of contraction. And after that, our muscle should go again to the relaxation phase. The first step which we have during relaxation is the termination of rapid nerve signals which are propagated along the motor neuron. When the nerve signal stops, there is no additional release of acetylcholine. So the acetylcholine, which is present in our synaptic cleft, start to become hydrolyzed by the enzyme which we call it acetylcholine esterase. The acetylcholine esterase receptor, the acetylcholine receptor become closed, and both the end plate potential at the motor end plate and the action potential in our sarcolemma start to decrease and decrease. Calcium channels which we have in our triad, both in T-tubules and in sarcoplasmic reticulum, return back to normal. And we have some calcium pump in the wall of sarcoplasmic reticulum, which utilizes ATP and bring back the calcium ion back again to the sarcoplasmic reticulum and store it there. The troponin again return to its original shape when we don't have any calcium ion. When troponin go back to its original shape, the tropomyosin come and cover the active site of actin. So we don't have any connection between actin and myosin anymore. And the muscle contraction stop and our muscle go to completely relaxation. These are the happening which we have during muscle contraction and muscle relaxation. You have many videos in your PowerPoint and also in this folder on your Blackboard, so don't forget to check these videos. Now I want to show you the other video which can show you the neuromuscular junction. Let me bring it here for you.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholinesterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
Okay, this is the happening which we have in the neuromuscular junction. And the next video which I want to show you is about the excitation contraction coupling.
Sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the myosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin-binding sites on the actin, allowing myosin heads to bind and form crossbridges. The crossbridges play a role in muscle contraction.
Okay, this is the other video which shows you how the actin and myosin can slide over each other and can make contraction.